So here we have a generic chemical synapse, and here is the presynaptic cell or presynaptic neuron communicating with the postsynaptic cell. Which could be another neuron or a muscle or gland effector cell. And today we are going to be focusing on these receptors here, the postsynaptic receptors, which receive the signal, the transmitter, they bind the transmitter that diffuses across the cleft that is released from the presynaptic neuron. So generically, postsynaptic receptors are integral membrane proteins. So they span the lipid bilayer. And this means that they have hydrophobic regions as part of their subunits to remain in the lipid bilayer. And they have an extracellular site for ligand binding, and this ligand is a transmitter at the chemical synapse. So some transmitter comes along and binds to the ligand, or binds to the ligand binding site on this postsynaptic receptor's extracellular site. And this ligand binding causes an effector function of some sort. So the postsynaptic receptor is the one that carries out an effector function, whether it be gating ion channels, indirectly or directly, causing hyperpolarization or depolarization of the membrane. All of this depends on the postsynaptic receptor and not the ligand or the transmitter binding. You may hear that glutamate is excitatory, but this is not necessarily always the case. Glutamate being a common transmitter, it commonly has an excitatory effect because it commonly binds to postsynaptic receptors that cause excitation or cause depolarization. The effector function depends on the postsynaptic receptor and not the particular transmitter that binds to it. The same transmitter can bind to different postsynaptic receptors that have excitatory or inhibitory effects. And the same postsynaptic receptor can have excitatory or inhibitory effects depending on the relative ion concentrations across the membrane. So even one postsynaptic receptor can have different effector functions depending on the context. So there are two major types of postsynaptic receptors, which is the main part of this video. These are ionotropic and metabotropic receptors, and there are many subtypes in each of these. Ionotropic receptors are a little bit more simple, and they're what you typically first talk about when you talk about postsynaptic receptors. So you have that ligand binding site, which all postsynaptic receptors have, that we're talking about today, and you also have that integral membrane protein. But this integral membrane protein also contains a channel pore. So when the ligand binds, the same macromolecule has a channel pore that it can open or close to conduct ion flux. So in this way, ionotropic receptors directly control ion flux. And direct means it's the same macromolecule, same protein that has the channel pore that can conduct ion flux and that binds the ligand. And for this reason, ionotropic receptors are also called receptor channels. And this name is helpful because it just says the two pieces. So you have the receptor that binds the ligand, and then also the channel, the ion channel piece of it, and it's just in one, they're combined. And these can be either excitatory or inhibitory. right? Ion channels can conduct ion current in both directions, and they can lead to depolarization or hyperpolarization depending on the relative ion concentrations. It depends less on the ion channel itself than the electrochemical driving forces driving the particular ions across through that channel. And contrary to some myths, these ionotropic receptors mediate all types of behaviors in the brain. So they're not just mediating simple reflexes, they mediate complex cognition as well. And a key attribute of them is that they underlie fast signaling. This whole process of gating the ion channel directly from in the same macromolecule, the ligand binding causes a conformational change that changes the channel pore, the orientation of those amino acids, the constriction. This doesn't take that much time, so often you see ionotropic receptors controlling or underlying the fast portions of signals in the brain. So now we're going to switch over to metabotropic receptors, and these indirectly control ion flux. So you have the metabotropic receptor here, and that binds the ligand just like all postsynaptic receptors do. They have the ligand binding site, and they carry out some effector function. What is different about metabotropic receptors is that the effector function is outside of the particular macromolecule. So this is one protein, and this protein can go on to bind to all sorts of different other proteins. Sometimes these signaling cascades get complex, and we'll see some examples later on. But it activates maybe phosphorylation of ion channels, and it can indirectly gate ion flux in that way. And it doesn't even these receptors don't even always act on ion channels. They can act on gene expression. They can upregulate or downregulate the expression of certain genes, which can underline long-term memory. They can phosphorylate different substrate proteins that aren't ion channels, but they could be ion channels. So there are a lot of more possibilities, complexities surrounding metabotropic receptors, and they can often initiate many signaling cascades. So they indirectly control ion flux, and again, indirectly means different macromolecule that conducts ion flux as binds the ligand. And maybe this is a little bit too simplistic of just comparing or contrasting that with this, is that it doesn't only control ion channels, it can control a lot of different other things. And as you can imagine, this is also can be excitatory or inhibitory. And it could also be modulatory in a way that doesn't directly depolarize or hyperpolarize the membrane, but it could lead to changes in the intrinsic excitability of the neuron. It could result in changes in the resting potential from which all other signaling occurs. So it may not even be directly excitatory or inhibitory. And these underlie slower modulatory signals, which themselves underlie modulation of behavior in just a very broad sense. So modulation of behavior includes stuff like changing emotional states, changing attention. So you can do change in attention, emotional states, different things like that. So there are two major families of metabotropic receptors. The most common talked about type of metabotropic receptor is the G protein coupled receptor family, or GPCRs. And in this receptor family, you have the G protein coupled receptor, which is typically a serpentine receptor. Serpentine receptor is just a cool, fancy name for a receptor with 7 subunits or 7 pieces. And you can tell the transmitter binds right there. And then this macromolecule doesn't have a channel pore like ionotropic receptors. Instead, when the transmitter binds, it results in a conformational change that allows it to bind to a subunit of a protein called G protein. It binds to the alpha subunit, whereas this is the beta gamma complex. It binds to this alpha subunit, activating this alpha subunit, and that alpha subunit can then go on to initiate other signaling cascades. It can commonly results in the phosphorylation of other proteins through something called the CAMP pathway. Often, just generically, metabotropic receptors signaling cascades result in the production of a second messenger. And a second messenger is simply a small, like freely diffusible metabolite with signaling capabilities. So a very common example of this is CAMP, or cyclic AMP. And there are a whole bunch of other examples of second messengers, but these are produced by metabotropic receptor cascades. So in this case of CAMP, the receptor, the G protein coupled receptor, activates the alpha subunit, which then activates a different enzyme that produces CAMP. And then CAMP can go on to activate another protein or enzyme that can phosphorylate its own target proteins. And that is a very long chain of events. So yes, these signaling cascades can get really complicated. But here, there are many sorts of effector functions. Here, we're showing the gating of an ion channel. This can happen. There can also be different effector functions that metabotropic receptors, or GPCRs, activate. The other category of metabotropic receptors are the receptor tyrosine kinases, or RTKs. Typically, although not always, these receptors, metabotropic receptors, live as monomers when they're inactivated, and then they combine to become dimers. So here you have two of the monomers combine to become dimers when the transmitter binds. So these are the little transmitter binding, and then this, just like the GPCR cascade, can initiate other second messenger signaling cascades, which could result in ion channel gating or other effector functions like changes in gene expression, changes in the localization of proteins. For instance, instead of gating ion channels, these cascades could result in a whole new channel being inserted into the membrane. Here is just one example of a second messenger cascade getting pretty complicated. This is a hydrolysis of phospholipids cascade that starts with a G protein coupled receptor. Let's see if we can make this bigger. And this is just an example of the complexity. I'm not going to go super into depth about this, and if this is your first time seeing it, you're not expected to understand this completely. But It starts with a G protein coupled receptor that activates a G protein, which activates another enzyme that creates second messengers. IP3 is a second messenger, DAG is a second messenger, calcium can be a second messenger as well. And this is just an example of how these signaling cascades can be really complex, and they can also branch off. So, for instance, this enzyme cleaves this PIP2, which is a phospholipid in the bilayer, into two different second messenger cascades. So this means that this cascade branches off into two cascades. So a lot of complexity here, and this is just one of the examples of how metabotropic receptors can get a little bit out of control, activate a whole bunch of stuff, and resulting in modulation of behaviors, slower signals, changes in the intrinsic excitability of neurons, basically like plasticity, learning, memory, all of the buzzwords in neuroscience, a lot of them are linked to metabotropic reception. So those are the two major types of postsynaptic receptors: ionotropic receptors, which we didn't talk about too much in length because they're relatively straightforward, and metabotropic receptors, which can get a little bit out of control. They're doing a whole bunch of different things. But yeah, hope that helps, and see you next time.